Good afternoon, everyone. Thank you for being here on this rainy day. As mentioned, I'm Marianne Pohl, and I'm a Director of Alliance Management at ALEX. And today I'm going to talk to you about end-to-end AI applications for advanced antibody engineering and multispecifics design. But first, I want to tell you a little bit about the company that I work for, ALEX. We are a biologics innovation company, and we are dedicated to developing advanced biotherapeutics using AI-powered solutions. We're a subsidiary of Crystal Pi, and we've grown to over 100 employees across 3 different countries. And from a researcher perspective, we have a ratio of about 2-to-1 wet lab biologists to AI computational scientists. And I want to highlight this because I think it shows that we've really built our team fit for purpose to integrate AI and ML into traditional wet lab capabilities. But all of our scientists, wet lab, dry lab, work together in the same 30,000 square foot lab space. And together, they have built a platform for discovery and engineering based on 3 AI engines, which I'll talk more about later. And we use these AI engines to focus on 3 core applications. And since 2019, we've worked with over 30 different partners in biotech and pharma. We also have an internal pipeline of programs, and some of our partnership highlights include licensing agreements with Johnson & Johnson and UCB. And most recently, just last week, we entered into a strategic partnership with Lilly for bispecific antibody development. So that's been very exciting. Now I'd like to tell you a little bit about our tech stack. So this nice image here is showing the different levels of capabilities that we have. So on the bottom here, you see the traditional wet lab infrastructure. And at ALEX, we have all of the standard bells and whistles you come to expect from a state-of-the-art antibody discovery and engineering lab. So just from that perspective, we could compete as a preclinical discovery and development company. But where we differ is how we've been able to integrate AI and ML, and that's what you're seeing on the middle level of the diagram. So all of this is based on a database that we have curated called the Atlas database. And we have spent a considerable amount of time and resources in generating data for this database and curating it. And it includes primary sequence data from antibodies and antibody-antigen pairs, also data around antibody-antigen interactions, so data around affinity, epitope recognition, and complex structures. And then we also have data around developability metrics, so things like thermal stability, hydrophobicity, polyreactivity. And like I said, this database consists mostly of internal data, but we've also curated data from publicly available data sets as well to try to make it as comprehensive as possible. And so using the data in our Atlas database, we've been able to develop 3 AI engines that are really the core of our in silico technology. And they fall into 2 different categories, either a folding module or design modules. So our folding module is called CrystalFold, and it is an in silico crystal structure prediction platform to look at antibody-antigen interactions or protein-protein interactions. And in order to use CrystalFold, you only need the primary amino acid sequence of your antibody and antigen, and it will generate this virtual complex prediction. And so when we generate these complex predictions, we can then use the learnings from that to move into downstream design and engineering efforts, and that's where the design modules come in. So ZenProt is our generative AI platform. It's a large language model that we developed specifically for antibodies and proteins. And then we have Zentient, which is our predictive AI suite. So this consists of an array of algorithms that we've developed for predicting different features of antibodies, including things like binding, affinity, and developability properties like thermostability. So using these 3 platforms, we can move from the folding prediction to design and then into actually producing molecules and testing them. So we all know there's a lot of talk about AI and a lot of hype around AI in biologics discovery. And at ALUX, we like to take a pragmatic approach to this. And so in thinking about our AI tech stack, we wanted to apply this technology to challenges that we think that this tech is best suited to overcome. And so with that, we have decided to focus on 3 flagship applications, one being hard targets. So that includes things like GPCRs, ion channels, targets where it's been difficult to generate antibodies to using traditional or standard methods. We also focus on advanced engineering, so that is multi-parametric optimization, and I'll give you an example of that today. So trying to optimize multiple features of a molecule in parallel. It can also include things like pH sensitivity engineering or engineering in species cross-reactivity. And then finally, we have multispecifics design. As we all know, this is a direction our field is going. And so how can we use our AI to design the best multispecific molecules in these complex formats? So that is what we're focusing on at ALUX. And I'll give you a little bit more information on how we're approaching these 3 applications. So the first is hit identification for hard targets. So as I mentioned, things like GPCRs, ion channels, peptide MHC complexes, it's either difficult to make the soluble antigen to do an immunization campaign, or in the case of peptide MHCs, you can immunize, but most of the antibodies you get will be just to the MHC and not the specific peptide MHC complex. So using our AI folding and design modules, we can do things like AI design to stabilize an antigen. So that's what I'm showing you at the top here. This is a multipass transmembrane protein. And what we were able to do is maintain the ECD in its native conformation, but introduce large-scale mutations into the transmembrane region. So we're talking tens, dozens of mutations that would really be nearly impossible to do using traditional rational design approaches to stabilize the antigen so that we can produce a soluble, stable form that can be used for immunization. Alternatively, or in parallel, we can also do AI-guided epitope focusing. So in this case, what we're doing is de-immunizing the protein scaffold. So for the example here with a peptide MHC complex, the majority of the MHC protein might represent the mouse MHC paralog, while the epitope of interest, which is where the peptide is binding, would represent the human protein. And so this goes beyond just standard chimeric like sequence grafting, but all in all, this is another approach that we can use to generate a stable antigen. So using these approaches, hopefully we generate a robust immune response, and we then move into hit identification. So we can do all the standard hit identification, B-cell sorting, phage display, NGS analysis, but where we're able to use AI is in rare hit identification and expansion. So by analyzing the NGS data using our AI tools, we can identify rare clonotypes within the repertoire that may be just a singleton. There's only one copy of that particular clone in the repertoire, but it happens to be a really functional, highly desirable antibody. The other thing that we can do, which I think is pretty cool, is structure-guided hit amplification. So let's say we've identified an antibody from a campaign that has the function that we're looking for. We can use that as a seed antibody to go back and interrogate the repertoire using our AI tools. And it will allow us to identify other clones within the repertoire that are predicted to have similar function to our original clone, but are dissimilar at the sequence level, where they share commonality as they're predicted to have the same structure. So all in all, these approaches allow for us to identify novel epitopes, identify better functional binders, and also enhance IP protection. As I mentioned, our second core application is advanced engineering. And so, as you know, when you're trying to design a molecule that's going to make it to the clinic, you need it to have optimal properties in terms of things like affinity, specificity, low immunogenicity, and a good developability profile. And so in the past, using rational design or mutagenesis approaches like with a display library, you can optimize one to maybe a few criteria, properties all in parallel, but it's very labor intensive and it's really not an efficient process. At ALUX, we've been able to employ our AI technology to optimize up to 7 properties of a molecule in parallel. With just a few design tests and express cycles, we're able to achieve our target product profile for given molecules. And so that's what I'm showing you in this graph on the right. We start with the parental antibody in the lower left corner, and with just a few rounds of optimization, we can improve properties of the molecule and achieve the TPP, like I mentioned, and I'll talk more about this later. And then finally, our third focus is multispecifics design. And so one feature of this is AI optimization of SCFEs for bi and multispecifics. With our platform, we can do high throughput conversion of Fabs to SCFEs, and at the same time ensure that these molecules maintain binding and optimal developability profiles. And then on the bottom here is our common light chain platform, and this has been of particular interest with many of our partners. And the nice thing about our CLC platform is there are no transgenic mice required, and we can start either from 2 given antibodies to target A and target B, and engineer them, or we can start from scratch and employ some approaches in a discovery campaign to identify CLCs quite early on in the discovery process. So those are our 2 main areas that we focus on in multispecific design. So now that I've given you a high-level overview of what we do at ALUX, I want to give you a few examples of how we've been able to put this tech into use. The first being an example of protein engineering with GP130. I'll talk about AI optimization of SCFEs, and then finally our CLC, common light chain engineering platform. So for the GP130 engineering, as many of you might be aware, the IL-6 trans signaling pathway is a known pathway to target for autoimmune diseases like IBD. And in this IL-6 trans signaling, it's mediated by the IL-6, IL-6R complex interacting with GP130 on the cell surface. GP130 also interacts with IL-11, and with this cytokine interaction, this pathway is involved in immune regeneration. And so from a therapeutic perspective, you would want to block the pro-inflammatory pathway mediated by IL-6, 6R interaction, but spare the regenerative pathway. And in terms of the therapeutic landscape with this trans signaling pathway, there is one molecule in the clinic. It's a GP130 Fc fusion decoy receptor. However, it's not specific to IL-6. It also blocks the IL-11 interaction. So in terms of our protein design goals, we wanted to improve the affinity of GP130 to the IL-6, 6R complex as much as possible, and decrease the affinity to IL-11, IL-11R. And of course, the goal here is to enhance efficacy, to tamp down the pro-inflammatory pathway, but spare the tissue regeneration. Another important aspect is maintaining developability. We want a profile that's on par with the wild-type GP130 protein. And then, of course, from a business perspective, we need patentability and FTO to move this molecule forward. So this is how we approach this particular challenge. And as I mentioned previously, it starts with protein structure predictions using crystal fold. So we generate these complex predictions for GP130 and its antigens, and this allows us to have some insight onto which residues we can play with in terms of optimization, and that moves into the generative AI part of the process using XenProt. And so this generates thousands of sequences. So in order to triage those, we move into the pre-screening phase where we use predictive AI to down-select a small number of hits or clones or variants that we want to express and test in the wet lab. So we do this whole cycle, and based on the results from the first round of wet lab testing, the data that we obtain from that can then be used to further train and tweak our pre-trained models for this specific project. And so in this particular example, we did 4 rounds of optimization, expressed around 230 variants, and we're able to identify 7 GP130 variants that showed a 3-fold increase in affinity, which is shown on the right here, and a 20-fold decrease in affinity to the IL-11 complex. So this whole process is pretty efficient, and within just a few rounds of optimization, we were able to achieve our selectivity and affinity goals. And while it's not really shown here, we're also considering developability in the down selection during the in silico screening. So this is just a little bit more detail on this same case study. On the left here, I'm showing you the output of the crystal fold complex structure prediction of GP130 with IL-11. And I believe that this particular complex, the crystal structure had not been published when we started the program, but as we were working on it, it was published, and we compared the PDB structure to our in silico prediction, and the DOTQ and RMSD scores are shown here, which are quite good, and so we consider it a successful prediction, and it further validates our crystal fold model. And then on the right here is a dot plot showing the affinities to IL-11 and IL-6 of our different variants. So what you can see here is all the dots in gray are the first round of optimization. So you're not seeing a huge improvement in affinity with just the one round, but as you move into the second and especially in the third or fourth rounds, which are in purple and green, you start to see a big gain in affinity and a big decrease in affinity to IL-11. And each of these variants had about 2 to 6 mutations. And so it really shows that using the data we generate from the early rounds of optimization can really help train the model to get to our desired profile in the later rounds of engineering. So what I showed you was a protein engineering case study, but of course we can apply this same general approach to antibody engineering and affinity maturation. And so I just wanted to share this quick case study with you as an example of that. So in this particular example, we had an antibody that had an affinity of 5.78 nanomolar, and we generated a complex prediction and used that for our in silico design and down selection. And with just 2 rounds of optimization, we expressed 65 variants total, and in only 5 weeks' time, we were able to obtain a 13-fold increase in affinity compared to the parental molecule, so sub-nanomolar affinity gains. And on the right here is a dot plot just showing you the affinities between round 1 and round 2. So now I'd like to move into another example of our capabilities using AI for SCFE optimization. And so, as I'm sure many of you are painfully aware of in this room, SCFE conversion can be a real bottleneck for bispecifics design. So you've done this hit campaign, you have a panel of binders that are functional, they're great, and then you go to convert them to SCFEs and you lose activity or binding or function. And this is exactly what happened in this particular example. So we had 11 SCFEs that we had converted from canonical IgG format. And 3 of those 11 failed to express at all, including our best functional binders. So obviously this is a problem. So here's how we approach this at ALEX. But first we had to think about the cause of the issue. And so with SCFEs, there can be several reasons why you lose expression or binding or activity. A lot of it can be due to mispairing, so you're losing the interface between the heavy and light chain domains. You can have issues with linker breakage or misorientation or even dimerization. And so we have developed specific tools to enhance the interaction between both the CDRs and the frameworks to try to avoid this problem. So how do we approach this? Well, it's a similar general cycle as the previous case study I just shared. It's structure prediction, generative design, in silico down selection, and testing. So in this particular case, we performed the crystal fold analysis, and of those 3 SCFEs that didn't express, and we're able to identify the critical residues that are involved in binding to the antigen. So we want to spare those paratope residues so that we maintain binding. And then when we move into the generative design step, we can be a little bit specific on the design conditions, so like the number of mutations, the amount of sequence diversity, or the number of variants that we want to design for this particular round of sequence generation. And then from there, we can move into the virtual screening, so we're filtering based on things like structural integrity, solubility, stability, and potential sequence liabilities. So all of this can be done rather quickly. The in silico design is done in less than 1 week, and the wet lab burden is quite low. We're expressing less than 10 SCFE variants per parental molecule. And here are the results. So on the top left, we're looking at the percent monomer of these variant SCFEs. As you can see, the parental SCFEs did not express at all, so that's why they're 0, but all of our variants are showing expression levels that are on par with the parental IgG. And we see that also with the yield, which is shown in the bottom graph on the left. But of course, affinity is also very important because it's one thing to be able to produce a molecule, but if it doesn't bind, then who cares, right? So when we look at affinity of these SCFE variants, we see that they are on par or within a 5-fold range of affinity to the parental IgG. So all in all, this is a pretty rapid way of quickly solving this issue of Fab to SCFE conversion. But that's only one step. If we want to take it farther and move into multiparametric optimization, we can do that by just doing a few more rounds of optimization. And with that, we can improve properties like affinity. As I mentioned before, we can engineer in species cross-reactivity and also look at developability properties like thermostability, removing PTM, risk sites, immunogenicity, et cetera. So this can be just dealing with the expression and affinity issue can be done in a very short amount of time, but we can take it farther depending on the needs of the program. And so that goes back to this graph I showed you earlier. All in all, with this general approach that we've developed, we can achieve our TPPs through this rapid active learning by applying the data from successive rounds of optimization to tweak our AI models. And so, as I mentioned, there are a lot of parameters that we can look at, some of which are shown here on this slide. And then finally, I want to touch briefly on our CLC engineering platform. As I mentioned, this has been a really exciting development for us and has been of great interest to our partners. I think this slide is deceptively simple because there's a lot that goes into engineering these CLCs, but I just wanted to give you a general overview and show you an example of where we're starting with 2 existing antibodies to 2 different targets. So in this particular case, we had antibodies to target X and Y, which bind at about an affinity of 0.1 nanomolar. So in order to perform CLC identification, we use a few different approaches in combination, which include structural modeling, yeast display, and NGS. And using this sort of proprietary approach that we've developed, we have been able to, in this case, we were able to identify 10 CLCs. But we didn't stop there because these weren't optimal in terms of affinity and developability profile. So then we can move into the next phase, which is using yeast display, generative design, and virtual screening to really optimize the molecule so that we've improved affinity. So in this case, a 4-fold improvement in affinity to target X while maintaining affinity to target Y, and big gains on the developability side, including really lowering the polyreactivity of the molecule and ensuring good expression. So with that, I'll just wrap up and summarize. In terms of our 3 different flagship applications with hard targets, AI-guided antigen design and immune repertoire mining have really enabled us to perform epitope-focused discovery and identify more functional antibodies. Second, in terms of multiparametric engineering, synergistically integrating and fine-tuning our AI models has really allowed us to reduce the number of design cycles and minimize the wet lab expression burden. And then finally, I think the cornerstone of our work is accurate modeling of protein complex structures, and it's really essential for our downstream multiparametric optimization approaches. So with that, I'll end. I just want to say I'm here for the rest of the conference, along with my colleagues Kyle and Yvonne, if you'd like to learn more about ALUX and talk to us about any potential partnership opportunities. We also have 2 posters downstairs, B8 and B9, so feel free to stop by and say hello. And with that, I just want to say thank you very much for your time, and I'm happy to take any questions that you may have.
I think we don't have time for questions, so perhaps people will find you at the booth to ask questions. And there is information about the next keynote